In today's video, we'll be learning about the regulation of respiration. So our breathing occurs by two mechanisms. One is the autonomic mechanism which happens itself, and that's why we can breathe without even thinking. Whereas there is also the voluntary responses or the voluntary mechanisms which acts when we are conscious of our breathing and when we want to control our breathing. Predominantly, as we all know, breathing is an autonomic process, and this is regulated mainly at three levels. The first one is the neural level or the neural level of regulation, and then we have the chemical level of regulation, and finally there are some reflexes which control our respiration. So here I have shown a diagram, and here we have our pons and our medulla. These are the two areas where the respiratory centers are present. So let's just name our respiratory centers. In the pons we have the pneumotaxic center. In medulla we have two centers. We actually have three centers. I will get to the third one later. One is the dorsal respiratory group, and the other one is the ventral respiratory group, which is located ventrally in the medulla. So these are the two groups. In addition to that, we also have the Preboard Zinger nucleus. Anyway, what these respiratory centers do is that the centers from the pons and medulla, they will somehow influence the spinal cord or the anterior horn cells present in the spinal cord, and these spinal cords, the neurons starting from the spinal cord, they will go and they will innervate the respiratory muscles. Which are the respiratory muscles? The respiratory muscles are one, that is the diaphragm, and the second respiratory muscle are the intercostal muscles. So this is the pathway. So these two muscles are stimulated, and when these muscles are stimulated actively, what happens is that inspiration occurs and there is inflation of the lung. What about expiration? What we need to understand is that expiration is a passive process. It does not require a neural stimulus. It's just like stretching a spring. We need energy to stretch the spring, but the recoil of this spring happens spontaneously. In a similar manner, expiration is a passive process. So now let's just write down the centers for neural control. As we have written now, there are centers in medulla and there are centers in pons. In the pons, we have two areas. One is actually the pneumotaxic center. We have the apneustic center. Now the apneustic center is still controversial and about its existence, and therefore we will not discuss about it in this video. When we come to the medulla, the centers are the dorsal respiratory group, the ventral respiratory group as we have discussed, and in addition to that we have the Preboard Zinger complex. So now let's discuss the role of each of this center in our respiration. Let's begin with Preboard Zinger complex. Now what is the Preboard Zinger complex? Let's say this is our Preboard Zinger complex. Now what does the Preboard Zinger complex do? Well, the Preboard Zinger complex contains the pacemaker cells. Just like we have the SA node in the heart, which discharges rhythm towards the heart or sends down the electrical impulse which will ultimately guide the cardiac cycle. In a similar way, the respiratory cycle of inspiration and expiration is guided by spontaneously, involuntarily, autonomically by the Preboard Zinger complex. So these pacemaker cells, they will go and they will stimulate the anterior horn cell of the spinal cord, and they will go and stimulate or end at the muscles of respiration, which I have said earlier, which are the diaphragm and the intercostal muscles. Now this signal which the Preboard Zinger complex actually sends, it is actually known as the inspiratory ramp signal. As I have said earlier, we only require neural signals for inspiration. We do not require a signal for expiration, as expiration is a passive process. Now there is a speciality of these signals which is sent by the Preboard Zinger complex. The Preboard Zinger complex do not give out. What happens is that these discharges will increase gradually for 2 seconds, and after 2 seconds they will suddenly stop. For the next 3 seconds there is no signal. What it means is that inspiration occurs for the first 2 seconds, and then there is a gap of 3 seconds for expiration to occur. Then a new involuntary discharge is sent by these pacemaker cells. Again it will rise for the first 2 seconds, then it will stop for the next 3 seconds, during which expiration occurs, and so on. So this is how we breathe usually. Our quiet breathing is happening all the time through this mechanism. That is, the pacemaker cells, they send the inspiratory ramp signals to the anterior horn cells of spinal cord, ultimately stimulating the muscles of respiration, leading to inspiration. What is the role of the other centers? So we can say that this is what happens during quiet breathing. Now, what is the role of the other centers, such as the dorsal respiratory group, the ventral respiratory group, and the pontine centers? Let's see. Now, for every respiratory center that we are going to discuss, we are going to use this format. First is the location of the nucleus. For the dorsal respiratory group, it's actually not the name, it's location. So for the dorsal respiratory group, this is actually present, as the name suggests, dorsally in the medulla, and it is present at the level of nucleus tractus solitarius, which is actually a nucleus which is present in the medulla, corresponding to the responses from glossopharyngeal and the vagus nerves. You'll learn more about this in neuroanatomy. The next question is, what type of neurons do they contain? Do they contain inspiratory neurons or do they contain expiratory neurons? The answer is that DRG contains inspiratory neurons only. There are no expiratory neurons. Now you might ask, as I've said in the beginning, that expiration is a passive process. So why do we have or why do we need expiratory neurons? Well, the reason is that normally expiration is a passive process, but during forced breathing, there is forced inspiration and there is also forced expiration. Forced expiration is an active process involving a lot of the muscles. It even includes the abdominal muscles. So for forced expiration, we do need the role of expiratory neurons. However, DRG only contains inspiratory neurons. And this is because DRG is also involved in quiet respiration or in our normal day-to-day breathing activities. Now coming to the role of the dorsal respiratory group, what happens is that the inspiratory neurons in the dorsal respiratory group, they receive afferents, as I have shown in this previous figure. This is our dorsal respiratory group. Like the Preboard Zinger nucleus, which do not receive afferents from any part of the body, the dorsal respiratory group, they receive afferent from the baroreceptors in our body. Secondly, from the chemoreceptors. We'll discuss later about the chemoreceptors. And they also receive afferent fibers from the stretch receptors in the lungs, in the bronchus, and so on. And according to these responses, they will modulate the function. They will project into the Preboard Zinger complex, and then they will modulate the discharge from the pacemaker cells. So depending upon the responses from the baroreceptors, which will identify the pressure changes in the blood, the chemoreceptors will identify the changes in oxygen levels, carbon dioxide and hydrogen ions in the blood, and the stretch receptors of the lung, they will identify the changes of inflation and deflation happening in the lung. And all of these changes, they will be carried to the dorsal respiratory group by the 9th and 10th cranial nerve, and the dorsal respiratory group, in response to that will send down neurons to the pacemaker cells and modify the response of pacemaker cells. In addition to that, that is their first afferent response. In addition to that, these dorsal respiratory neurons directly themselves will go and synapse with the anterior horn cells and affect the muscles of respiration. So that is the role of dorsal respiratory groups. So if we have to write down, they receive afferents from the sites which we have mentioned earlier, and the inspiratory neurons they will project to the Preboard Zinger complex and modify its response. The other response is directly on the anterior horn cell of the spinal cord, which will then ultimately affect the respiratory muscles, and they modulate the inspiration. And this modulation occurs during normal quiet respiration. What about forced expiration and forced inspiration, such as in the case of heavy exercise? That is where our ventral respiratory group comes in. Just like the ventral respiratory group, as the name suggests, it is located ventrally, and it is located between the nucleus ambiguous and the nucleus retroambiguous. These terms are a bit difficult to remember, but we learn about them in neuroanatomy. So these two nuclei are present in the medulla, in the ventral part of medulla. So between them, we have this ventral respiratory group of neurons. Now they are involved in forced inspiration and forced expiration, or in forced breathing. And therefore, as I've said, expiration is an active process in forced breathing. Therefore, they include both inspiratory and expiratory neurons. What do? These inspiratory and expiratory neurons, they will stimulate the anterior horn cell of the spinal cord, and the mechanism is same. This will go and stimulate the muscles of respiration. They will also include the accessory muscles of respiration as well. That is important. The accessory muscles of respiration, other than the diaphragm, intercostal muscles, they are also stimulated, leading to forced breathing. That's the role of the medullary centers, and we end with the ventral respiratory group. Now we move to the pontine center. As I've said, I'm not going to discuss about the apneistic center here. We'll be rather discussing about the pneumotaxic center. Now the name of the nucleus, once again, as I've said, it's a difficult name. We have the nucleus parabrachialis, and we have the collicular nucleus. So these are the two nuclei of the pneumotaxic center. Now the type is, it contains both inspiratory and expiratory neurons. Basically, what the pneumotaxic center does is that the role of pneumotaxic center is that it will switch off the inspiratory ramp, or it will switch off the inspiratory signal. And how it does is that this is our pneumotaxic center in the pons, and this pneumotaxic center, it will send down inhibitory signals to the dorsal respiratory group of neurons, and that will switch off. As I've said, dorsal respiratory group is involved in the quiet breathing, so this will switch off the inspiratory ramp signal. Now the question is, what happens if the pneumotaxic center is destroyed? What happens is that we have said that the inspiratory signal goes up for 2 seconds, right? And then it stops, then we have a 3-second pause, then a new signal comes in, and so on, right? What happens if the pneumotaxic center do not, or it's inactive? In that case, what happens is that this inspiratory signal will be prolonged. There will be prolonged inspiratory signal. So instead of 2 seconds, you may have 4 or 5-second inspiratory, the signal will increase. What does this mean? This means that our breathing rate will reduce. There will be a decrease in the breathing rate, because this is one breathing cycle, including an inspiratory signal of 2 seconds and a 3 seconds of pause. That is one cycle. Now here, when the pneumotaxic center is cut off, our breathing cycle's duration has increased. We are taking deeper breaths, and therefore our breathing rate will reduce. On the other hand, when the pneumotaxic center is hyperactive, what will happen? Well, the opposite will happen. What happens is that the inspiratory ramp will be switched off easily, and therefore we will start to hyperventilate, or there will be increased rate of breathing. So that's about the neural centers. Now the next topic, I hope you have understood the neural regulation. It's not just the neural centers which control our respiration. Our respiratory rate is also affected by the changes of carbon dioxide, hydrogen ions, and oxygen in the blood and CSF. That's what we are going to explain next. Now, chemical molecules, how do they affect respiration? Well, they affect at 2 levels. One is at the central level, at the central nervous system, and then they affect via the peripheral level, or through the peripheral chemoreceptors. So we have central chemoreceptors which can identify these changes, and we also have the peripheral chemoreceptors. The central chemoreceptors, they identify changes in the carbon dioxide and hydrogen ion levels. The peripheral chemoreceptors, they will identify the changes in carbon dioxide, oxygen, and hydrogen. But the speciality here is that oxygen levels in the blood is detected by the peripheral chemoreceptors, not by the central chemoreceptors. So oxygen does not act at the level of central chemoreceptors. It acts its effect at the level of peripheral chemoreceptors. Now, what are the factors which stimulate respiration? The factors which will increase the respiratory rate are: one is increase in the carbon dioxide levels in the blood and CSF. Then we have the increase in the hydrogen ions and a decrease in the oxygen level. So these 3 conditions, increase in carbon dioxide, decrease in oxygen, and increase in hydrogen ions, these are the 3 conditions which will stimulate or increase the rate of respiration. Now let's go into detail about chemical regulation. We start with the central chemoreceptors. Where are they located? They are actually located in this area, that is the chemosensitive area. This area is known as the chemosensitive area, and it is present at the ventral part of medulla. What happens in this area? To understand that, we'll just look at the effect of carbon dioxide and hydrogen ions. As I have said earlier, they respond to changes in hydrogen ions and carbon dioxide. So we have here 4 scenarios, and let's analyze what happens in each of these conditions. The first scenario is increase in hydrogen ions in the CSF. When the hydrogen ions increase in the CSF, there will be direct effect on the chemosensitive area, and this chemosensitive area, they will control the respiration. They will send signals to increase the respiration. So respiratory rate is increased. That is the effect of hydrogen ions in the CSF. What if the hydrogen ions are high in blood, not in the CSF? What happens then? Well, the reason is that there is a blood-brain barrier. If the H plus ions are higher in the blood, they cannot cross into the CSF or affect the chemosensitive area because of the blood-brain barrier. So due to the blood-brain barrier, which we write as BBB, there is no effect, or there is a minimal effect of rise in hydrogen ions in the blood to the respiratory center. Therefore, the increase in H plus ions in the blood does not have much of an effect on the central chemoreceptors. Now let's take the third scenario. What happens when the carbon dioxide levels increase in the CSF? When the carbon dioxide levels increase in the CSF, there will be a direct effect of the carbon dioxide, just like the hydrogen ions, and they will increase the rate of respiration. Now we have to ask a question. Why is respiration increasing when carbon dioxide increases or H plus ion increases? Why not decrease? Well, in biochemistry, you will learn that the increase in carbon dioxide or H plus ions, that will lead to a condition called as acidosis. And the response of the body to acidosis, one of the compensatory mechanism is to wash out the excess carbon dioxide. The carbon dioxide and hydrogen ions are linked. That is a separate discussion which we will deal in biochemistry, not in this video. But what we need to understand is that our body needs to wash out this excess carbon dioxide to prevent acidosis. They need to wash out this excess H plus ions, and that is done by increasing the rate of respiration so that we wash out the excess carbon dioxide. So that's the effect of CO2 in CSF. Now what if instead of the CSF, if there is an increase in the blood? Well, unlike the H plus ions which cannot cross the blood-brain barrier, CO2 can cross the blood-brain barrier. And then what happens? Therefore, it can affect the chemosensitive area. This carbon dioxide, it will combine with water. It will form a bicarbonate which will dissociate into H plus ions and the bicarbonate. So what happens? This H plus ions, now they are not in the blood, they are out of the blood, they have crossed the blood-brain barrier. So just like the effect on CSF, they will directly act on the chemosensitive area. So that is how the increase in carbon dioxide levels in the blood will affect the central chemoreceptors indirectly, not the direct effect of CO2, but first by converting into H plus ions, and then these H plus ions will act on the chemosensitive area, and that will increase the respiratory rate. Now a question is, I have said that oxygen does not affect the central chemoreceptors much. Why? Why don't there be a stimulation of the central chemoreceptors when the oxygen levels are low in the blood? The reason is that is because of the hemoglobin buffer system. Even if the oxygen levels are low in the blood, even when the PO2 is low in the blood, the brain and the chemosensitive areas and the autonomic centers in the brain, they will still get adequate blood supply and oxygen supply. And that is because of the hemoglobin buffer system. Therefore, unless the oxygen levels are extremely low, unless the PO2 levels are extremely low, the central chemoreceptors are not affected because they still get adequate oxygen due to the hemoglobin buffer system. So with that, we now move into the part of peripheral chemoreceptors. So I have said that the peripheral chemoreceptors, they will respond to all three stimulus, that is increase in carbon dioxide in the blood, increase in H plus ions, and decrease in oxygen level. Now, where are they located? Well, they are located at two sites. The first one which we have shown here near the arch of aorta, they are known as the aortic bodies. And the next one is here at the bifurcation of the carotid arteries, that is known as the carotid body. So these are the two bodies which are responsible, or these are the two peripheral chemoreceptors, and these peripheral chemoreceptors, they will detect the changes of these three molecules, and then they will stimulate or send neurons into the dorsal respiratory group. This is what we said, dorsal respiratory group receives its afferents from the peripheral chemoreceptors, and they send it through the 9th and 10th cranial nerve. The arch of aorta, the aortic bodies will send through the cranial nerve 9, which is the glossopharyngeal body. The carotid bodies will send through the cranial nerve 10, which is the vagus, and they both will go and affect the dorsal respiratory group and make the changes in the discharge, leading to an increase in respiratory rate. Now we move into the part of reflexes, and the only reflex which I will mention in this video, there are a number of reflexes happening, but one important one which we will be discussing is the Herring-Brewer reflex. Now this is a two-part reflex, that is, it's called as the Herring-Brewer inflation reflex, and there is also a Herring-Brewer deflation reflex. Basically what happens is that when the lung expands, in inflation reflex what happens is that when the lung expands, the walls of the bronchi contain a lot of stretch receptors. So these stretch receptors get activated, and these stretch receptors they have the afferent neurons. They are connected to the vagus nerve or the cranial nerve 10, and through the vagus nerve they will go and stimulate or alter the discharge from the dorsal respiratory group, and this will stop the inspiratory signal. Why is this done so? Well, the reason is very obvious. This is to prevent the overinflation of lung. This is a reflex mechanism in the body to prevent the overinflation of lung. So when the lung inflation reaches a maximum, the stretch receptors are stimulated, they will stop the inspiratory signal coming from the dorsal respiratory group. So that's about the inflation reflex, and an exactly opposite or vice versa happens in case of the deflation reflex. That's about what we discuss in this video about the regulation of respiration. And finally, I will end this video with something called as the Ondine curse. Now, Ondine's curse is based on a historical story. Here what happens is that the person's autonomic control or autonomic neural control is disrupted, of regulation of respiration is disrupted. What does this mean? This means that the normal breathing which happens involuntarily of a person stops. As a result, the person must consciously breathe always. How is the voluntary control occurring? The voluntary control occurs from our cerebral cortex. The cerebral cortex will directly through the corticospinal tract, they will affect the discharges on the anterior onset of spinal cord, and through the anterior onset of spinal cord, they will control the muscles of respiration. This will be the only mechanism which will be possible in Ondine's curse. In other words, if the patient sleeps, the breathing of the patient will stop and the person may die. So that's it for this video.